Hello everyone, my name is Huiyan. I'm an assistant professor in biomedical engineering program at the University of Guelph. So today I will present our recent work on multiplexed extracellular vesicle protein detection using a innovative dot blotting technique. So here is the outline of my presentation. First, I will introduce why we do this work, the motivation of this work, what are the EVs and the application of EVs for cancer management. Then we will introduce this new technology inspired by the conventional protein dot blotting technique. We call it it's for highly sensitive multiplexed EV protein detection. In the end, I will give conclusions and talk about the outlook of this work. So extracellular vesicles, also called EVs, is a relatively new research field, but recently become a very hot topic for biologists and biomedical engineers. So basically, EVs are small vesicles in the nanometer range, secreted from basically all the cells, normal cells, cancer cells, plant cells, other animal cells into body circulation. So we can find the EVs in most body fluids such as blood, urine, saliva, and so on. And compared to the other types of biomarkers such as proteins, or some circulating tumor cells or mRNA, DNAs, they are more robust and quite abundant in the blood plasma. So become very interesting potential biomarkers. And more importantly, these small vesicles can carry molecular cargoes such as proteins and nucleic acids that are very similar to their cells of origin. And these small molecules function in intercellular communications. So imagine that if we want to analyze some cells from a solid tumor in the body, conventionally we need to do a surgery, which is called tissue biopsy, take some cells or tissues and do the downstream molecular analysis. But apparently this type of surgery is invasive for some patients with other diseases and can cause some complications. So if we can detect these EVs which carry very similar molecular cargo as their cells of the tumors from the blood samples, just do this analysis from blood test, that could be much less invasive. So that's the motivation of the work for measuring the proteins in these EVs from the blood plasma samples. So as I said, actually EVs are quite new research field, and there's a lot of challenges. One major challenge is because of their small size. Each EV is around 50 to maybe 200 nanometers, called exosomes, or maybe under 1 micron usually. So they are much smaller than a human cell, which is typically about 10 micron. So on average, it's about 100 nanometer for human EVs. And until today, there's a lack of preparation and analytical techniques for highly sensitive detection of the EV molecular cargoes, and for multiplex analysis. It's still a very young research field, and the existing test equipment and assays are of high cost and need some specialized skills to operate. So still need to develop some technologies for EV sample preparation and protein analysis. So here is one kind of standard method for current EV isolation from complex fluids such as blood plasma. We will use this technique for the results section. So I will introduce this technique here. It's called ozone column, which is basically a size exclusion chromatography, and they make a commercial product with this ozone column, and it can isolate the EV vesicles with the size larger than 70 nanometers. So as you can see here, they said that it can separate more than 99% of the proteins and separated from the EV vesicles. So it's quite standard method, but still is of a bit high cost, and all these isolation or purification method could cause the loss of EVs. Basically, just depending on the level of the cost, but all these isolation method could cause some loss of the EVs. So imagine if we want to analyze very small amount of EVs. If we do the isolation, it takes more sample, it has more cost, and could lose some EVs. So here we are trying to develop a method without the isolation step, just directly measure the EVs from raw blood plasma sample. So here is the workflow of the procedure. Is quite similar to the. It basically is use the same membrane, nitrocellulose membrane, with the conventional dot blotting technique. So here we have a nitrocellulose paper membrane, and we pipette a spot of the capture antibody, which is anti-CD63, which is a common EV surface protein. So by this way, after blocking, we incubate with the EV samples. Could be the isoned EVs, could be the raw blood plasma samples. So through the antibody, the EV molecules in the samples will be captured and concentrated, enriched on the membrane surface, and also inside the membrane 3D matrix structure. Then we wash the membrane and spot array, microarray of the detection antibody. Each detection antibody has one row, and the target is one EV surface protein marker. So through this, we can measure the EV surface proteins, and this antibodies has fluorophores in it, so that we can use fluorescence-based detection for imaging. So another innovation in this work is the use of the gold nanoparticles. So we use gold nanoparticles to enhance the local surface plasma resonance, so that the fluorescence signals can be enhanced for highly sensitive EV protein detection.
The last person is going now.
Sorry?
Yeah.
Hello? Can you hear me? Okay. Then the signals will be imaged with a fluorescence microscope. So through this way, we don't have to pre-isolate EVs from the sample, and we can do multiplexed EV protein detection where there's antibody microarrays on the surface. So it's similar to the conventional dot blotting, but used for EV applications and allowed highly sensitive multiplex EV protein detection. So our first consideration is which type of membranes we should use. There are many different types of membranes: cellulose membrane, nitrocellulose, PVDF membrane. All these membranes are actually used for protein detection, antibody-based protein detection. So we assume this should be compatible for the EV protein analysis. So here we compare the cellulose and the nitrocellulose membranes. They have a bit different surface chemistry. The nitrocellulose membrane has more negatively charged surface groups. And here another difference is their pore size. The average pore size for the cellulose membrane is 11 micron, and the nitrocellulose is about 400, 500 nanometer. And imagine the average size of the EV is only 100 nanometer. So EV is much smaller than the pore size of those type of the membranes. So they can go inside the membrane matrix and get trapped there. So here we take a CD9 protein as an example and measure it on the EV surface, and we are trying to compare the two types of the membranes. So we have the isoned EV. Remember, we can isolated EVs from the blood plasma sample using the size exclusion chromatography, or we can measure from the raw sample without any EV isolation step, just the blood plasma, and also it's a black control. So for all these three samples, the natural cellulose membrane works a little bit better, provides a little bit higher fluorescence signals. That's why we choose this natural cellulose membranes other than the cellulose membrane. So here the higher sensitivity could be from the higher antibody binding capacity, because we use fluorescent labeled antibodies to test the antibody binding. We found that the nitrocellulose membrane has a bit higher antibody binding, and also it results in a little bit smaller spots compared to the cellulose membrane. Basically, the cellulose membrane is used for many filtering applications, and the solution can really spread quickly in that membrane, maybe because of the pore size difference and different surface charge and so on. So with the smaller spots, the signals can be more concentrated, so it can provide a stronger signal. So here we choose the natural cellulose membrane, which is actually commonly used for Western blotting. So here is the scanning electron microscope images to characterize the EVs enriched on the natural cellulose membrane and the membrane without EV as a control. So on the right side is the membrane without the EV. As you can see, it's quite porous. Here's the scale bar, 2 micron. And here we have the EVs as circled. These are the EVs. As you can see here, some EVs were actually cracked. Like here, probably because of the dehydration process. Before the ICM imaging, we used the ethanol alcohol with a gradient of concentration to gradually dehydrated for the imaging process. So here is a cracked EVs, but they were captured on the membrane using an antibody targeting the EV surface protein. And the surface protein is common for all the EVs. So basically all types of the EVs from the blood sample will be captured on the membrane. Then the next step is to optimize the blocking protocol. We do this because we found that the background signal is still high compared to the specific IC signal. So maybe we think, the conventional method is, the standard is to use 1% BSA, bovine serum albumin, which is a animal protein, to block the membrane surface where there's no antibodies, so that it could prevent the binding of the EVs onto the other area of the membrane, so that ideally the EVs only bind to the specific antibody that can reduce the background. Then here we use a blank sample without any EVs, and we compare the background signal with different concentrations of the BSA, and we want to choose a better one. Actually, the better one has a lower signal because this is blank sample. We want to have a low background signal or blank signal. So we found that the 1%, 5% BSA, they're similar, not statistically different, but with 10% BSA is much better. And also here shows the signal to noise ratio of the different concentration of BSA. So from here we choose to use 10% BSA for the blocking step. Now another important factor to optimize is the antibody concentration. So if the antibody concentration is too low, it's not enough to provide strong signals. But if it's too high, there could be something similar to the hook effect in conventional immunoassays. Actually, too many antibody molecules, it could block the specific binding between the antibodies and antigens. So actually the signal will be lower. So here we're trying to find the appropriate antibody concentrations. So we use 3 concentrations: 10 microgram per ml, 20 microgram per ml, and 40 microgram gram per mil. Same here, we tried isoned sample EV samples, meaning the EVs are pre-isolated from blood. Non-isoned is just a raw blood plasma sample and a blank sample. And we found that 20 microgram per mil of the detection antibody, the one in the middle, gives the highest signal. And here is showing some image of our microarrays. Here is to detect the CD9 protein on the EV surface. So that's why we choose 20 microgram per mil as the appropriate antibody concentration for the following procedures. Then next we said we want to do some metal enhanced fluorescence to improve the sensitivity of the assay. So here we use the gold nanoparticles for enhanced fluorescence signals. If for the metal enhanced fluorescence, the critical point is the distance between the gold surface, metal surface, and the fluorophores. Usually the distance should be around 5 to 90 nanometer, because if they are too close to each other, actually the fluorescence signals will be quenched by gold. And if they are too far, of course, there's no enhancement effect from the gold surface. So although we cannot exactly measure the distance, we will try to compare between different concentrations of the gold nanoparticles and the size of these gold nanoparticles to get an appropriate distance range. Then after some comparison and optimization, we found that 5 nanometer nanoparticles with this particle concentration gave the highest signal. So that's what we choose for this step. And here the EV concentration in this experiment is actually only about 10 to the 5 orders of magnitude in EVs per milliliter, which is quite low, because the goal is to improve the signals from the low concentration of the EV samples. So here we choose a relatively low EV concentration to test the gold nanoparticles. So here, as you can see, with the gold nanoparticles, the signals were improved compared to the ones without gold nanoparticles. And with the blank sample, they were quite similar because there were minimal fluorescence signals, and of course there were minimal enhancement. So that's the benefit of the approach. The gold nanoparticle only enhance the specific signals, but has minimal effect of the blank signals. That's why if we do a standard curve, it will help enhance the assay sensitivity, because the curve will pick up more quickly. Another concern is whether the gold nanoparticles can be distributed evenly in the membrane. So if they are not evenly distributed, it will affect the repeatability of the assay. So here we show that intraday and interday variations were all below 15%, which is the DFA guideline for developing a diagnostic test. So we think the repeatability is high for this measurement, for this type of the assay. So after these optimizations, we try to do some multiplex EV protein detection from only 6 microliter of the cancer EV spec human plasma. So, typically with the isolated, like the isone column, at least we need 500 microliter of the samples to isolate the EVs and then to detect the EVs. So using our method without isolation, we save the samples by at least 100 times. So we were detecting 3 markers, CD9, CD24, and APCAMA, 2 common cancer markers, which can be found in many different types of cancer. And here is the standard curve for these EV proteins, and also we did a ELISA for the CD9 just to compare with the results from our assays. So the red plot is for the ELISA conventional method, and here we see that from our method with the metal enhanced fluorescence with the dot blotting, the curve pick up more quickly, showing our method is more sensitive compared to the conventional gold standard ELISA. Okay, so here just a small summary of the advantage of this technique. Only requires 6 microliter of the blood plasma samples. We don't need to pre-isolate EVs, so it can save the cost, save the reagent, and more importantly, it will avoid the loss of EVs. Also, we can do multiplex protein analysis in a microarray format. The assay is scalable if we want to measure. So here we show that we measure 3 markers, but if we want to do 5 markers, 10 markers, 20 markers, we can print more proteins in the microarray, and it will not affect the existing antibodies in the microarray. So we found up to 4 orders of magnitude more sensitive than conventional ELISA. And also it's a low cost method because you know that bioreagents are pretty expensive, the antibodies, but here we only need a very small volume of the antibodies, about 1 nanoliter per spot. So it's much smaller volume compared to conventional assays, so it lowers the cost of the assay. Okay, to conclude, I would like to thank my collaborator, Dr. Chen, Joshua. They helped fabricate the gold nanoparticles. My grad student, Nika and Gisela Roshan, for their hard work, and we thank the financial support to finish this work. Okay, thank you very much for your attention, and I would like to take any questions.